Okay, so we start now. I give you a template. This is one of the complex we are going to draw. So before we draw the complex molecule, we will try to construct the DNA molecule. Okay. So before we start, we have to connect our project. Go to change directory. I hope by this time you all know how to do it. Our directory is in our desktop, so I just connect to DNA. Open our project, which is already there in the name of DNA. Then I go to project again, open the project table. Anyway, we don't need the project table now. I just clear the workspace and we're going to draw this. Now see, we're going to construct the DNA, all right, based on the template that is given here. Okay, how we do? You can see for drawing this, we need the template which is there under fragment here. So when you click the option fragment, it will open up a lot of fragments and then go to the second last option, which is again known as fragments. When you click that, under this you can see there are several options. All right, first of all you need to click grow, then choose the option double strand aDNA. You can see it is showing A chain grows 5' to 3. Okay, so here also you can see 5 to 3, right? So after that you need to draw the double stranded DNA structure following the sequence. You can see here CA is there, C, 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 C 5 times. So we click this C, G options 5 times. 1. After clicking one time, you can see it shows 2 arrows here. That means the other structure is going to be built above this. So since there are 5 times CG, so we are going to click 4 more times. Already 1 time is there. 1, 2, 3, 4. Then after that, there is A, T, A, T again. That means this one, A, T, then T, A, then again A, T, then again T, A, then GC, GC. Then again 1, 2, 3, 4. 4 times CG. 1, 2, 3, 4. Our template is constructed already, you can see. Alright. Now after constructing, you can cancel this window and then go to the option ribbon and select show ribbon for all residues. If you all feel that you are unable to see my screen or listen to me, instead of texting, you just reply to me, okay? Because I have minimized the window. If you text me, I may not be able to see. So immediately, if you have any doubt, you just talk to me. Unmute your speaker and just speak to me. All right. So far, any doubt? No doubt. Very good. Now, after drawing this, we need to prepare this molecule, minimize this this giant molecule, or we can say macromolecule or DNA structure. Okay, we need to enable minimize this to get it into a proper shape. For that, we go to the option Prep Wizard. You can see the symbol here. You know already what is the purpose of it. We need to click it and wait for a while. You can see another window has popped up, protein preparation wizard. In this, generally we know there are 3 steps, right? Import, process, review and modify, and then final step is refine. Now in this case, all the steps are generally used for the preparation of any protein like receptor or enzymes. But in this case, we have not imported any structure. We have constructed a DNA molecules. So we have to avoid first 2 stages. We directly go to the stage refine. Okay. Generally, what happens from where we import the structure? We import the structure from PDB. Correct. Here we have not imported, so we don't have to review the structure, analyze the structure, and modify anything. We just go to refine and then click the last option, that is minimize. Okay, you see it has given a warning. This type of job must have one entry included in the workspace. Now I would like to ask you, anyone, please answer me why this warning has come.
Must create entry
Can
first.
Farida answer this? Why this warning has come? Yes, what is that?
Must create entry first.
Create entry to where? Yes. Create entry to where?
Workspace.
Uh, okay, Rajakumari said create entry. Now Farida, can you say create entry to where? From where to where?
Workspace. From workspace.
From the
Hello,
workspace?
sir.
Yes. Yes, I can hear you. From the workspace to?
Project table.
Very good. So after we draw the molecule, we cannot perform any other job until we create an entry of this particular molecule to project table, correct? That's why it is shown the warning. So what we have to do before minimizing the structure, we need to create entry. You see the option here, create entry, right? How about Saja? Do you agree? Saja, we create entry to
Yes, correct,
good.
Kevin.
Now you can see the structure name we have given DNA. Okay, it appear in project table, right? So once we have created entry to project table, now we can perform any other job, right? So this is again one of the thumb rule for any structure that you draw in this workspace. So after drawing, compulsorily you have to create entry to the project table. Then only you can perform any job, right? Then again we go to the option preparation wizard. You can see and the another window has come, right? Then what we have to do here? Which step we have to go? There are 3 steps, right? Import, preprocess, review, modify, and refine. Which step we have to go?
Define.
Which one?
Define.
Yes, tell me. Refine. Okay, in
Define.
refine,
Define.
what we have to click?
Minimize.
Minimize, right? Very good. Now see, when we click minimize, it doesn't show any warning, right? Now you can see it is showing the running restraint minimization. So after clicking, if you are not sure whether the job is still running or it has finished, how do you check? How to know the
Go
job
to
is still
task.
running?
Go to task.
Where?
Monitor job.
Monitor job
Task.
under where? Task, right?
Task.
Okay. In my version, it is given as application. So under application, in your version, advanced version, it is mentioned as task. So you can see a
Task.
monitor job at the end, right? So when you click monitor job, it will show the status whether the job is running or finished. You can see what it is showing. It shows finished, right?
Finish.
Okay. That means our preparation is finished. So DNA preparation is finished. So you can close this window. You can see earlier there was only one entry in the project table, right? Now you can see there are two entry in the project table. That means what? Moment it prepares the raw protein, it will create another entry into the project table, which is our prepared protein compared to the raw protein, or you can say the prepared DNA structure. Sorry, prepared DNA structure compared to our raw DNA structure that we have constructed earlier. All right. Now, after construction of the DNA structure, what is our next step? What is the next step? Generally, we do. Yes. Can anyone tell me?
Docking.
No. Before docking, we follow one step. Generally, you see, after we do ligand preparation, then we do protein preparation. Protein could be anything like receptor or enzyme. After preparation of the target, or we can say in other words the target, here the target is DNA, right? After target preparation, we do not go to docking status directly. We follow one more step. What is that?
Generate state.
Generate state. No. Think. What are the steps that you followed?
Grid generation.
Very good. Grid generation, right?
Mm.
The grid generation, what you can tell in other word?
Preparing the site.
What is that?
Preparing the active site.
Uh, Not really. We cannot say preparing the active site.
Mm-hmm.
Can you use the correct word and see every word has different meaning, right? Different meaning. So you have to use the correct word to define it. What is that step you go?
Covering the space that contains the, the receptor site.
What is that? Yes, once again.
The receptor site, or where the drug, the active site.
Active site what? What do you call that steps? Yes, what you said grid generation is okay, correct. But other than grid generation, what actually you do in grid generation? What is the purpose of grid generation? Grid is nothing but a 3D box, right? It's a 3D box. Where you prepare the 3D box and what you call to that step. Try to recall and tell me. Or you can apply your own logic and tell me.
I think I already applied my logic.
Still you apply. What about others?
Okay.
Rajakumari?
Is it to identify the binding site?
You are a bit closer. Yes, almost right. See, now there are two things. You all said binding site. One thing is common. Yes, something related to binding site. Now you said identify the binding site. Is it really identify the binding site or something else? You are very close actually. How about if I say assignment of binding site? Assigning the binding site. Which word do you think is correct? Identify the binding site is correct or assigning the binding site is correct? Yes, tell me.
Assigning the binding site.
Why do you think assigning the binding site is correct?
Identify, because assigning is like you are determining the active site rather than you identify where the active site is.
Yes. Actually, we can identify like where the active site is, correct? But identifying doesn't mean never may not mean that we are also assigning that site as a binding site where our molecule should go and bind, right? So before docking studies, we need to assign the binding site in the target so that the molecule should go and test itself whether they are able to bind into that particular binding site or not, right? That's why after preparation of the target could be generally we call target as a protein, right? Since they are amino acid in nature, most of the cases the target will be receptor enzyme. But in this case, it is what? It is DNA, right? So we know the drug molecule never go and bind to each and every part of the DNA or each and every part of the target, right? So first we need to identify the binding site and we assign the binding site surrounding that area. So that during docking the molecules can go and bind to that particular site. Okay. So we are going to do the grid generation. Grid generation is nothing but creating a 3D box, right? Creating a 3D box surrounding the binding site, which we also call it as a assigning binding site. So grid generation is nothing but assigning binding site in the target. Let us see how to do. So in this case, we go to application, okay, go to glide and receptor grid generation. Okay. Generally, if you remember, can you tell me what generally we do? Generally in a binding site assignment, if you remember, we uh, we tend to pick any one part of the molecule and then go to site, right? Yes or not? But in this case, generally in other cases, what happens? In other cases, in the receptor, inside there will be a co-crystal structure, right? And we try to pick the co-crystal structure, identify, and you can see a green structure. Then we go to the option site and create the binding site. But in this case, since there is no co-crystal structure inside the DNA molecule, right? Okay, we follow a little different procedure where we go to the site first. Instead of clicking any part of the molecule, we go to the option site. Okay. After going to the option site, since you see earlier we used to use the first option, that is centroid of workspace ligand, right? Yes or not? If you remember. But now there is no ligand inside. So we cannot assign a binding site surrounding the ligand, for which we used to use the first option, centroid of workspace ligand, okay, selected in the receptor tab. So we use the second option, that is centroid of selected residues. In this option, after clicking highlighting this option, you go to the option specify residue. Okay, click this, it will appear, it will show another window like this, active site residues. Once you see this, now go to the workspace in the DNA molecule. You know our binding site is located after 5. If you look at the template here, C, 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 right? The fifth C, start from fifth C to G, C, A, T, A, T, G. This is the binding site, right? So if you count in the DNA from here, 1, 2, 3, 4, 5. The binding site start from here, right? So we will click somewhere nearby here. You can click any portion here. Okay, see, moment we click here, it will show a 3D box. Are you able to see 3D box? Yes, you can see the 3D box here. Now generally after seeing this 3D box, which you call it as a binding site, okay, or a grid, we generally tend to give the name here and run. But in this case, we go to the option advanced setting. When you go to the option advanced setting, you can see it has given the coordinate size 10 Armstrong for X, Y, Z. And if you look at the structure here, inside there is another box appeared, green color, right? Actually, the binding site is surrounding that. And you see, it is not covering all the five, you know, nucleotide, that is C, A, T, A, T, G. It is covering only 2 to 3, 1, 2, 3, 4, that's all. So what we do, we increase the size of this green color box by increasing the coordinate at X, Y, Z. What we do, this length we can increase up to 10 to 20. See, the moment we are increasing, it is also increasing the size in the workspace. So we increase to 20. X, Y axis also we increase to 20, and Z axis also increase to 20. Now you can see in the workspace, it has covered almost most of the area. Is it clear? It has covered almost all the area of the binding site that is located here, CATATG. Alright, then click OK. After you click OK, then you give name here. We give the name here. For giving the name, what is the thumb rule? We just remove the numerical number and give the name. So you can give it as a DNA. Okay. And what is today's date? Today is 10th. We can give today's date as well. 10th, 7, 14, and then we click run. Now you can see once we click the option run, something is revolving surrounding the option run, right? So the moment you click here, it will show the status whether it is still running or finished. Otherwise, we have another option, that is we go to application or task, and we can see the monitored job where it will show the status it is running, and we have to wait until it finishes. Now all of you, do you have any doubt until this step? Speak.
No.
No doubt? Very good.
Mhm.
So you remember your task, right? Your case study. You have to construct a DNA structure like this based on the template that will be given by your professor, Prof. Ammu, right? And you will do the talking studies like this. How about Raja Kumari? Any doubt?
For DNA, no.
Very good. Excuse me, I have to attend a phone call. Hello. Hello. Yes. আরে বাপটু কিতা খবর খ আমি একটু অনলাইন ক্লাসও আচ্ছা খ কোনো ব্যাপার না ও ঠিক নি তো আমার বাড়িতে তুই কই আছ
My target also before.
Look at it here.
Hello all. Can you all hear me? Yes? Okay, good. Hmm. Let us check the status. Yeah, you can see the job is finished, right? Shall we take a break here? Shall we take a break for 10 minutes? Yes, I need a break. Uh, can we take a break? I
Okay.
actually got an emergency. Sorry for
Okay.
interruption. I got an emergency phone
Okay.
call actually. I just need 10 minutes break. We'll be back. Okay? Okay?
Okay.
Okay. Meanwhile, you discuss. You can open your Maestro. I can also see the Rajakumari's work and any other doubt if you will have. Okay? So I just stop here.